Okay, greetings all. Welcome to this session of the Algorithmic Thinking in Bioinformatics course. Today we will be looking at a clinically motivated question. How do we find the different types or subtypes of a disease? Okay, a disease, a complex disease specifically, such as autism spectrum disorder or cancer, for instance, doesn't come in one form or type, right? So there are different types and subtypes of cancer, and it is useful to know what are these different subtypes so that we can tailor the treatment and the diagnosis of these diseases according to the subtype of the disease in a particular patient who comes to the clinic, right? And so that is the clinical question we'll be looking at today. And we are going to ask, is there a data-driven and a computation-driven way to understand or identify the different subtypes of a disease? Okay. And the computational problem and the approach we are going to take is clustering, an unsupervised machine learning approach you may have seen in earlier machine learning courses in this program. And specifically, we will be looking at the k-means algorithm for clustering. You may have seen k-means in the earlier courses, but the emphasis here will be on designing new k-means algorithm variants using the notion of probabilistic modeling. And that I hope would be something new that adds on to what you may have learned about k-means in earlier lectures you may have, or earlier courses you may have been exposed to k-means clustering. Okay. So that is as a short intro to the session. So let's just jump right into it. Like I said, the clinical questions, we have looked at several sessions on several biological questions and their computational problems and algorithmic solutions. And today we'll be looking at this clinical question of how do we identify the different subtypes of a disease from data that has been measured from different individuals with these different disease subtypes. Okay, how do we group those patients into different categories so that each category has a certain subtype of this single disease that we are interested in? Okay, and what is the associated computational problem? Like I said, clustering and its algorithmic solution. Like I said, it will be using the k-means algorithm or heuristic, but the emphasis or the important part about it I wanted to emphasize is we'll be designing new variants of this algorithm by taking a probabilistic modeling view of k-means. Okay. Here is an example of a disease subtype. This is an example of a cancer, specifically leukemia. So this is a type of cancer that affects the blood cells and the blood forming tissues like the blood and the bone marrow, for instance. And knowing the specific subtype of cancer will tell us how to properly diagnose it and properly tailor the treatment to that particular subtype. Okay. And so we have data. So can we take a data-driven approach to identify the subtypes of this leukemia cancer? Okay. So what we have here is all the samples along the columns. So these are all the samples, right? And all the genes along the rows. Okay. So these are samples or individuals, right? So or patients from whom these samples are taken. Okay. And then from each of those samples, we have measured the activity or expression of different genes, right? So that if you take, for instance, this particular box over here, right? So this box is red in color, so red means high. Okay. And so this says that this last gene catalase, right? So this last gene catalase is expressed at a high level, is that activity is high in this particular individual, right? So there is this particular individual we are looking at. In this particular individual, right, we look all through here, right? I hope I got the same individual here. Yes. So in this particular individual, we have this expression of this gene catalase is high. But if you look at this other gene here, adenosine deaminase, that particular individual, right, has a medium expression, okay, or somewhat lower expression, comparatively lower expression of this particular gene. Okay, so that is what the data is. So at an I comma jth entry, right? So the column is J and the row is I. So it is the expression or activity of gene I in that particular individual J. Okay, and then you have these genes along the rows and individuals along the columns. And the question now is, we want to understand how these individuals may be grouped based on the measurement of the genes in these individuals, right? And here it is very obvious, as you can see here, that there are these two groups, right. So, there is one group of individuals, that is this first block, and then there is this other group of individuals, that is the second block, right. So, in this first group of individuals, right, so that is what is shown here. So, this is subtype 1, right, and this is subtype 2, right. So, all of these individuals here have this first set of genes which are very high, red in color, and then the second set of genes which are very low. And then in subtype 2, where the rest of the individuals, right, you have the second set of genes are very high, right, but then the first set of genes are very low, right. So, that is what you can see here, that using the expression or the activity of these genes in these different individuals, it is very clear that there are two subtypes of this disease, and those subtypes I am just labeling them ST1 and ST2, but it so happens that this form of grouping actually corresponds to the known subtypes of leukemia. So, here we are looking at this blood cell related cancer called leukemia. So, there are two types of leukemia, ALL and AML. That depends on which particular blood cell is most affected. And so, this grouping without knowing any of the labels, right, just looking at the data on the expression of these genes and then looking at the grouping of them into ALL and AML, into the subtypes 1 and subtype 2 corresponds to the ALL and AML, right. So, this grouping is so successful that if a new patient comes in and then you take their blood sample and then you do a gene expression measurement, and then you figure out among these set of genes which ones are high and which ones are low, then you can use that to figure out if that patient will belong to this subtype 1 ALL or subtype 2 AML, right. So, this is one of the examples of how do we identify the subtypes of a disease from gene expression data. Okay. Just wanted to clarify that in this previous slide, no labels of the individuals were given. So, it is an unsupervised setting. We are just looking at this data and then we are looking at, is there any patterns in the data, is there any clusters in the data, and we do find these clusters ST1 and ST2 clusters of individuals or patients, and these two clusters then later on when we just attach the labels, we can see that those clusters nicely correspond to the known subtypes of leukemia called ALL and AML. Okay. So, if you look at this particular task that we just illustrated using cancer, so this is essentially clustering, right. So, you identify certain similarities and dissimilarities among a set of objects that you want to cluster, and you want to cluster or group n objects into k groups. In the cancer example, k was 2, n was some set of individuals, the number of columns, right. So, we wanted to group those n columns into 2 groups in cancer, and in general you wanted to group n objects into k groups, right. And we do this all the time. Our human brains are very good at grouping objects, right. So, if you look at a particular room, then we know that there are certain sets of furnitures, and then there are certain other set of clothes and certain other food related items. So, we know identifying the different objects in a room, we know what are the groupings of those objects into different categories, and the grouping is done in almost an unconscious way, if you will, right. But how do we automate this process, right? So, we group objects based on their similarity, but how do we automate this process? And there are different ways of automating this process. And k-means clustering is one approach, that is a center or centroid based approach, where we identify for each group one particular representative point, data point, and then say that all the points that are closer to this data point will be assigned to that particular centroid or center, right. And there are other distance based approaches. You look at the distance between these different data points that you want to cluster, right. And then the general idea is that for data points within a cluster, you want them to be closer together, and the data points in different clusters, you want them to be farther apart, right. And there are other approaches such as spectral clustering as well. Okay. Those are some graph based algorithms for finding the clustering or the clusters or groups of a set of objects. Okay. And why have people, I mean, these are just very very small set of example approaches. There are many other approaches that people have taken to do clustering, but why is it popular? Why are people interested in developing different forms of clustering algorithms? The main reason is that you can look at it as fourfold. Okay, there are 4 reasons for this. It allows exploratory data analysis. it allows you to just look at the data and then see if there is any patterns or groupings or clusterings like we had in the cancer example again. So, this is a form of unsupervised machine learning. You just look at the data here, for instance, and then you just understand that there are these two groupings of these individuals that naturally pop up, right? And so, can we use the computer to find it in an automated fashion, right? So, our human eye can easily spot that there are these two groups of individuals, but can we develop algorithms to do that as well? And so, that allows you to explore the data in an unsupervised setting, right? And it has predictive power, like we said, even if you do not reveal the ALL on AML labels in the previous slide, the natural grouping corresponded to this actual subtype of this cancer, this leukemia cancer. And so, you can also predict for a new patient sample that comes in, right, based on the expression of genes in this new patients, you can predict which type of or subtype of cancer that patient has. So, it has predictive power, and then it has other things such as if you want to compress your data, then you do not have to store all the data points, you can just store only the centroids of each cluster. Centroid is a single data point that represents all the data points in a cluster, right, all the objects within a cluster. So, instead of storing n objects, you only store k objects, the k centers that are representative of the different groupings of these n objects into k groups, right. So, this allows lossy compression. It can reveal interesting outliers. So, some data points can be closer to the center of a cluster, and then some of them may be in the periphery of a cluster. So, that helps you reveal some anomalies in the data and some interesting outliers in the data. Okay. For all these reasons, it is a very popular data science task, very popular machine learning, unsupervised machine learning task, and that is what we will be using also to identify subtypes of a disease in this particular session. Okay. And like I said, you may have seen k-means clustering, or you will likely have seen it in the other courses you have seen in this program, right. So, what is distinctive about this lecture? So, what new concepts will you learn in this lecture about k-means clustering, right. So, we will start with a standard presentation of k-means clustering. So, we will present a non-probabilistic approach called the hard k-means clustering, and then transition to a probabilistic approach called the soft k-means clustering. But something interesting about this lecture that I find interesting in this, and I hope you do too, is that we will focus on viewing k-means clustering as an inference plus parameter learning task, right, based on a probabilistic model. It is called a mixture model, and that is used to model clustering problems. Okay. So, we have a probabilistic model called a mixture model, and we will look at how do you do inference calculations, probability calculations on this model, and then how do you learn the parameters of this model, and then as we learn that, we will see that we are essentially clustering the data if we do those. So, this connection between probabilistic mixture modeling and this clustering, this k-means clustering, will help us design new variants of k-means algorithm. Okay. So, that is the kind of the main takeaway I want you to get out of this lecture, on top of the k-means exposure that you may have already had. Okay. If you have not had a k-means algorithm exposure before, we will also go through it step by step, starting with hard k-means, going to some kind of a vanilla or a plain soft k-means, and then use this probabilistic connections to design new variants, okay, enhanced soft k-means. And the main idea for this lecture comes from this book, David J. McKay, Information Theory, Inference and Learning Algorithms. I would like to acknowledge the content in that book. So, some of the figures and the content in these slides are taken as is from the book, and they are cited as DJM in the bottom right of the slide where I use it. And then there are a few other sources, CMB, which is a pattern recognition and machine learning book, and then EZL, which is a set of lecture notes where we also use some content from here to explain certain concepts. Okay. Again, these are cited in the bottom right of these slides. Okay. So, let us just simplify our problem. Instead of looking at all of those 20 to 40 genes that we showed in this cancer example, let us focus on only 2 of those genes. Okay. We will call it gene A and gene B, and then we will just plot all of our individuals or patients. Each X here is a patient, and then their gene A's expression and gene B expression in this 2D plot, okay, scatter plot. So, we have about 40 individuals here, n equal to 40 data points or objects that we want to cluster over 2 dimensions. In our case, it is 2 genes. Okay. So, this is what we will focus on. We will use this as a running example in most of the content that we will look at in this session. Okay.